Hello everyone. Today let's understand the physiology of pancreas, how it is affected with the digestion and how it affects the digestion. Also, in this class we'll study about the diabetes mellitus. So, to begin with, pancreas has dual roles. It also acts as an exocrine gland and also an endocrine gland. Now, what is exocrine and endocrine? When it releases substances or enzymes through a duct into the particular part, that is called exocrine. And when it releases its substances or products or enzymes directly into the bloodstream, it is called endocrine. So first, let's understand the exocrine functions of the pancreas. For example, imagine a clover-shaped cell where uh, they're columnar cells, and these are called the acinar cells. It also has nuclei and it releases the pancreatic juice into the secretory acini, which are produced by the duct cells. And the secretory acini, and these are released into the intercalated duct and reaches the duct of Wirsung, where it joins the ampulla of water and opens into the second part of duodenum. It is the main pancreatic duct and it is guarded by the sphincter of Oddi. And then comes the accessory duct, which is the duct of Santerni, which also opens into the duodenum but a 2 millimeter higher than the duct of Wirsung. So these two releases the pancreatic juices that helps in the digestion of substances, which we'll be discussing it later. And the sphincter of Oddi, which is present at the opening of ampulla of water into the duodenum, controls the opening of this duct into the duodenum and thus regulates the release of pancreatic juices into the small intestine. So, for the key points, duct of Wirsung is considered as the main duct and accessory duct is the duct of Santerni. The Wirsung duct is opened into ampulla of water that opens into the duodenum and it is regulated by sphincter of Oddi, which helps in contraction and relaxation, thus helps in regulation of pancreatic juices. So the functions of exocrine gland is the release of digestive juices that helps in digestion of carbs, proteins, fats, digestion of food in the small intestine. And the function of endocrine gland is hormonal that regulates your insulin, glucagon, and so on, which we'll discuss later. So first, let's understand the endocrine function of the pancreas. Out of the whole pancreas, there are only 1 to 2% of pancreatic weight, that is the islet cells, or in other words, endocrine cells. These are approximately 0.5 to 1.5 million islets of Langerhans. This endocrine part consists of cells that secrete glucagon, insulin, somatostatin, and pancreatic polypeptide. And for these islets are divided into four types: alpha cells, which contribute to 15 to 20%, and they synthesize and secrete glucagon. Then comes beta cells, which is 70 to 80% of the IL cells, and it secretes and releases insulin. And 1 to 8% of delta cells synthesize somatostatin and gastrin in very small amounts, and 1 to 2% are the F cells, which secrete pancreatic polypeptide, which decreases the absorption of food from the GIT. So these cells are innervated by both parasympathetic, which is vagal, and sympathetic nerves, and can readily influence their secretory activity. First, let's understand the function of alpha cells that releases glucagon. Like I said, 15 to 20% of the total IL cells are alpha cells or A cells. So what is the main function? It helps in mobilizing glucose. It helps in making glucose by glycogenolysis, which is it breaks down liver glycogen into glucose, which is used by the body. It forms glucose by gluconeogenolysis. It is formed from amino acids and glycerols. This leads to sustained blood sugar rise, whereas glycogenolysis gives us rapid rise, immediate rise in the blood glucose. How? By inhibiting glycogen synthase and by activating CAMP formation. But in gluconeogenesis, it's like slow formation, regulated and steady formation of glucose from lactate, pyruvate, glycerol, amino acid. So it is slow but sustained blood sugar rise in the blood. It can last for hours and days. Glucagon is also a powerful lipolytic agent. That means it breaks down the fats and releases fatty acids into the circulation. So, in the liver, the excess of free fatty acids are converted into ketone bodies, that is acetoacetic acid, acetone, and beta hydroxybutyric acid. These are released into the circulation. Therefore, glucagon is catabolic in reaction. That means it releases energy. Like what is energy? ATP. It releases ATP. It also acts as a ketogen. Ketogenesis, that means liver converts fatty acids into ketone bodies. So overall, it has a catabolic role by releasing energy from stored sources. So it also stimulates the secretion of growth hormone and somatostatin by the delta cells. It thus increases in heart contraction strength. So overall, glucagon is a catabolic. It mobilizes energy from storage and releases energy for the use of body. Then comes insulin. So it was first discovered by Banting and Best in 1921. It is a small protein but consists of 51 amino acids. This arrangement of the amino acid is varied in species, so insulin of one animal is injected for prolonged period into another animal may lead to antigenic properties by producing antibodies. Normal fasting levels of insulin is 10 to 50 microunits per ml. Now let's understand the transport. Insulin binds with a circulating protein called syn albumin that has anti-insulin effect, and the half-life of this insulin in circulation is only 5 to 10 minutes. It is fixed to many tissues but not RBC, and most of the brain cells do not bind to it. So bra- insulin does not affect ventromedial nucleus of hypothalamus and kidney, GIT, and RBC directly. The insulin exerts its effect without entering the cell on which it acts. So, what 80% of it is metabolized by the liver and kidneys by hepatic glutathione insulin transhydrogenase, and 20% gets mobilized by the rest of the body tissues and destroyed by the enzyme insulin protease. Now let's understand the regulation of glucagon secretion. Regulation is how is it stimulated by and how is it inhibited. So, hypoglycemia. When there is low sugar, insulin is stimulated. When you have a protein rich meal, insulin is stimulated. When there are GI hormones like CCK, gastrin, GIP, insulin is stimulated. I'm so sorry, we are discussing about the glucagon regulation. It is stimulated by hypoglycemia, protein rich meal, GI hormones, stress, fasting, exercise, infection, sympathetic activity, cortisol, acetylcholine. And it is inhibited by hyperglycemia, high blood sugar, free fatty acids and ketone bodies, insulin, and somatostatin. Like we can see in the diagram, pancreas releases glucagon, which helps in conversion of glycogen into glucose into the liver, and the glucose is released into the bloodstream. Now that we understood the regulation of glucagon, let's understand the regulation of insulin. It is opposite. So, in hypoglycemia, glucagon is secreted. In hyperglycemia, insulin is stimulated and released. It is also released in amino acid and keto acids. These are the fat derivatives that is in protein rich in glucagon, substances that increases CAMP, beta adrenergic stimulation, parasympathetic, especially during feeding, GI hormones like GH, thyroid hormone, and it is inhibited by alpha adrenergic stimulation, sympathetic nerve activity. Now let's understand the actions of insulin on carbohydrate metabolism, fat metabolism, and protein metabolism. First, to begin with, on carbohydrates, insulin increases the glucose entry into the tissues like the skeletal muscle, cardiac and smooth muscle, adipose tissues, white blood cells, islands, pituitary fibroblasts, mammary glands, aorta, liver, and alpha cell of pancreatic islet. Glucose transport across the following cells does not require insulin. That means insulin does not play a role for the transportation of glucose to brain. Brain can metabolize glucose without insulin except the ventromedial nucleus of hypothalamus. Therefore, brain cannot withstand hypoglycemia. So, when there are low blood sugars, the brain cannot handle it. And insulin does not transport glucose also to kidney tubules, GIT, and RBC. This is the reason why in diabetic patients, that is insulin deficiency, glucose absorption from these tissues remains unaffected. You mostly hear islands have been disturbed, liver has been disturbed, and so on. So, insulin produces hypoglycemia by the following mechanisms. How? It increases peripheral utilization of glucose. That means the glucose that is in the circulation is transported to the muscles, eyes, pituitary, and so on. It increases glycolysis in muscles and adipose tissue. There it breaks down the glucose, promotes synthesis of glycogen by activating glycogen synthase and simultaneously inhibiting enzyme of glycogen breakdown. It also directly decreases the glucose output from the liver. So that is anti-gluconeogenic. Now let's understand the actions of insulin on fat metabolism. First, it promotes the synthesis of free fatty acids and triglycerides in muscles, adipose tissue, and liver. And how? It stimulates lipoprotein lipase, which breaks down circulating triglycerides. Insulin inhibits hormone sensitive lipase in adipose tissue, so decreases the release of FFA from stored triglycerides. Insulin can increase the uptake of ketone bodies into the muscle. So that means it reduces free fatty acid release. It increases ketone body use by the muscles. Now the protein metabolism. It promotes the synthesis of protein. It increases amino acid uptake, especially into the muscle. It breaks down the protein. It is reduced. That means the breakdown of protein is reduced, which is anabolic. So to other miscellaneous actions include insulin directly decreases the urea output from the liver and increases uptake of potassium and phosphate. Insulin increases potassium entry into the cell, thereby decreasing extracellular fluid. Overall, insulin is glycogenic, anti-gluconeogenic, anti-lipolytic, anti-ketotic. So it favors the storage of absorbed nutrients and is a hormone of energy storage or hormone of abundance. So we have read a lot or heard a lot about diabetes mellitus. What is diabetes mellitus? High blood sugar, but which is also known as hyperglycemia. But how is it causing high blood sugar? It is because there is insulin deficiency or lower insulin, or the insulin activity is disturbed. So diabetes mellitus, what does this term mean? Diabetes is like a siphon or running through, and mellitus is sugar. Therefore, diabetes mellitus is a clinical state which is associated with flow of sugar in urine. The first main reason for diabetes is insulin deficiency. The other secondary reason could also be dearrangement of glucostatic function of liver. What are the predisposing factors? That means who can be affected by? First, hereditary. This is very high chances. That means if your mother or father or is diabetic, there are high chances that you can be affected with diabetes. So you should be very mindful when you're consuming food and plan your lifestyle. Age common is with increasing age and in obese people. Now, what are the signs and symptoms? These are mainly due to hyperglycemia. The signs and symptoms we see are because of the increased blood sugars and decreased utilization of glucose. That means there is a lot of blood sugar in the blood, but it cannot be utilized by the muscles or the cells. So this is called starvation in the midst of plenty. So the first one, as usual, hyperglycemia, raised blood sugar. This leads to predisposing infections like boils, urinary tract infection, and next glycosuria, present of glucose in urine, polyuria, excessive urination, polydipsia, increased thirst, so dehydration, polyphagia, low glucose utilization like glucostat cells of ventromedial nucleus in the hypothalamus, that is the satiety center, results in no inhibition of lateral nucleus, which is the feeding center. So we always have that hunger feeling and eat a lot. And then loss of weight. 1 gram of glucose loss in urine causes loss of 4.1 kilocalories from the body. Others include ketonuria, poor resistance to infections due to protein depletion. So what are the tests done? Which is the glucose tolerance test. In normal, it is less than 115 milligram per deciliter, and then post meal, which is done 2 hours after meal, it should be less than 140 milligram per deciliter. Impaired tolerance or pre-diabetic stage is 115 to 125 milligram per deciliter in fasting, and 140 to 199 milligram per deciliter after a 2-hour meal. In diabetes, if the fasting levels are greater than 126 milligram per deciliter, or the post 2-hour post meal is greater than 200 milligram per deciliter, they come under the diabetes patients. Now, we have understood about hyperglycemia. Now it's also very important to study about the hypoglycemia, which is low blood sugar. If the blood sugars are below 60 milligram per deciliter, they are considered to be hypoglycemia, and it mainly affects the brain because insulin does not directly affect the brain. The bloodstream has to directly send it to the brain. So that's it. directly affects the brain. Early symptoms: polyphagia, nervousness, sweating, tremors, palpitations, headache, anxiety. In later stage, confusion, irritability, fatigue, difficulty walking, convulsions, coma, cardiorespiratory center involvement, which is life threatening. Severe hypoglycemia is life threatening, so immediate treatment is required. Now that we understood the functions of the endocrine part of the pancreas in detail, now let's understand the exocrine, which is the pancreatic juice that plays a crucial role in digestion. So let's understand the composition and functions of pancreatic juice. The daily secretion is about 1200 to 1500 ml, which is 1.2 to 1.5 ml, and it is alkaline in nature, that is 7.8 to 8.4 pH because of high HCO3 content. The specific gravity is 1010 to 1018. It neutralizes acidic chyme from the stomach. Like we understood, the pH of the juices released in stomach are acidic. So the chyme that is made in the stomach becomes acidic. So when it slides down to small intestine, it has to be neutralized, and this is done by the pancreatic juice. Now the electrolytes that are present are sodium, potassium, calcium, magnesium, zinc, which are cations, and anions like bicarbonate, which is highest as the secretion rate is highest, the chlorine, and traces of SO4 minus and HP4 minus. Electrolytic composition varies with rate of secretion. At high secretory rates, chloride concentration falls and bicarbonate concentration rises. And total concentration of these two always remains constant. And sodium and potassium concentration are almost identical with those of the plasma and are constant on changing rate of secretion. Now let's understand the digestive enzymes that are released by the pancreatic juice. First, pancreatic alpha amylase. Main function, it converts carbohydrates to maltose. It is stable in pH range of 4 to 11. The pancreatic lipase, which converts fats to fatty acids and glycerol esters. So its pH range is from 7 to 9. And pancreatic proteolytic enzymes, these are powerful protein splitting enzymes in the pancreatic juice and are secreted as inactive proenzymes. So these are inactive form in the beginning, which is trypsinogen, and it is activated by enterokinase, enteropeptase, that is secreted by duodenal mucosa, and trypsin that acts as an autocatalyst, and it is broken down to form trypsin. Trypsin is the most powerful proteolytic enzyme of the pancreatic juice. It hydrolyzes proteins to small polypeptides. Chymotrypsinogen, which is also the inactive form, it is activated by trypsin to form chymotrypsin. It digests protein to small polypeptides, ribonucleases, deoxyribonucleases. These split the nucleic acid of ribose and deoxyribose type into nucleotides. And comes trypsin inhibitor. It is secreted by the same SNR cells and the same time as the pancreatic enzymes, it protects the pancreas from autodigestion. So once the function of trypsin is completed, it has to be stopped. So the trypsin inhibitor is also produced by by the pancreas and stops the function so that it does not eat away the pancreas itself. So the regulation of the secretion of pancreatic juice. It is regulated by vagus nerve, secretin, CCK, etc. And that's it for today. Thank you.